everyone. In this video I am going to describe how we construct DNA replacement vectors by double joint PCR, and most of you have performed this process. You have constructed a DNA replacement vector, but you might not realize how the process works. So I'm going to go over that in this video. Okay, so for our project in spanning fall 2023 and spring 2024, we are trying to delete DNA intervals, specific DNA intervals from the genome of an SK3 strain. So this process of constructing a DNA replacement vector for use with an SK3 strain starts with getting genomic DNA from an SK3 strain. So the one we like to work with is called RDGR170.3. In the genotype, if you're interested in that, the genotype is called SK3, I guess I can say SK3 plus, RID minus, Moose51 delta bar plus, and it has the big A mating type. Now for this video, we don't need to worry about that, or that. We just need to know that it is an SK3 strain. It has the SK3 genotype. It is a spore killer. It is an SK3 type spore killer. So the process of isolating genomic DNA from this strain requires us to take some tissue, usually conidia, and transfer that conidia to a liquid culture medium. And we will incubate the conidia in the culture medium for maybe 24-48 hours, usually with shaking, usually at about 32 degrees Celsius. And then after 32 to 48 hours, what we will do is we will, the conidia will germinate and they'll grow and they'll form usually a glob of mycelia, so a glob of hyphae. And what we'll do is we'll isolate that glob, squish out the water, and freeze dry it. So extract all the water with a special machine called a lyophilizer, which will place the tissue under vacuum and suck out all the moisture while it's frozen. And that helps break up all the cell walls. And so once it's completely dry, what we can do, we can store it for a long time under dry conditions before we extract, before we isolate the genomic DNA. But after it's dry, what we can do is we can place that tissue, that dry tissue, in a small microcentrifuge tube, and we can grind up the tissue with this micro spatula here. So now we have this fine dust of mycelial tissue, and all the cell walls have been broken up. Once we have that, we can isolate the genomic DNA from that mycelial dust. And we usually use a kit to isolate the DNA from that dust. So a kit having columns that bind the DNA and solutions to help separate the DNA from all the other components in that mycelial dust. So once we have the DNA isolated, we can store that at minus 20 degrees Celsius, essentially indefinitely. So this is a microcentrifuge tube, and for those of you who have already isolated DNA from a fungus, from fungal tissue, you are probably storing your DNA, the DNA from those the strains that you isolated DNA from, in the freezer at minus 20 degrees Celsius in tubes like this, So this is a DNA solution, and in this case, this is the DNA from strain RDGR170.3. So when I say genomic DNA, what do I mean by that? Well, if we look in each nucleus of this strain, what we should see is 7 chromosomes of the Neurospora crassa genome, and the DNA, well, not in the nucleus, but in the DNA, in the tissue, there should be mitochondrial DNA in the mitochondria. So we've isolated all of that, and probably millions of copies of each of these when we isolated the DNA. So this tube contains millions of copies of all of these chromosomes probably broken up into smaller fragments, and millions of copies of the mitochondrial DNA. So we will consider all of this DNA to be the genomic DNA. And if you're curious, the genomic DNA, the length of the genome is approximately 40 million base pairs in Neurospora crassa. So we are interested in making DNA replacement vectors for DNA encoded within the SK3 region of the genome. And if you've watched some of the previous videos, especially for returning students, you know that the SK3 region is on chromosome 3 in SK3 strains, like RDGR170.3. So the SK3 region is about 3 million base pairs long. So it comprises much of chromosome 3, chromosome 3 being 6 million base pairs long in SK3 strain. So approximately 3 million base pairs comprises the SK3 region. So all of the genes SK3 requires to act as a spore killer, to act as a meiotic drive element, to selfishly transmit itself to the next generation in a biased manner, are found in this region right here. So let's look a little closer at that region. So we know that this region contains all of the important genes, but we don't know what all the important genes are. We know the SK3 resistance gene, the gene that allows it to resist its own spore-killing gene, RSK, is found at about position 300,000, So if this is chromosome 3, the centromere is before the 1 million base pair position, and then we have 2 million, 3 million, 4 million, and 5 million base pair positions along this chromosome. So the centromere is found to the left of the 1 million base pair point. So RSK, that resistance gene, is found around 300,000. Now, the interval we are interested in, and all of your projects are based on this interval, either subintervals of this or intervals to the left of the what we call the V350 interval. That's located at about 350,000 base pairs, or position 350,000 on chromosome 3 and in SK3 strain. So we're interested in this region right here. So we have the genomic DNA isolated for SK3, but we're really only interested in the DNA in this area right here. And what do I mean by interested? I mean interested in this area here for making our DNA replacement vectors. So, for example, let's say your goal is to construct vector 375, which the purpose of which this vector is to delete what we will call the vector 375 interval, the vector 375 interval. And this is a sub-interval of that V350 interval. And so to construct the DNA replacement vector, what we will need to do, and I've alluded to this in the previous video last week, what we will need to do is we will need to amplify 500 to 1,000 base pairs from of DNA that is found to the left of this interval. We'll need to amplify that by PCR, and we will need to do the same thing. We will need to do the same thing for 500 to 1,000 base pairs that is found to the right of this interval, that is found to the right of that interval. So in this case, vector 375, this is a real interval someone in the lab is working on. It comprises about 400 base pairs of chromosome 3 DNA, so about 400 base pairs. So this vector is being designed to replace these about 400 base pairs with a hygromycin resistance, antibiotic resistance gene. Okay, so two of the key steps are amplifying 500 to 1,000 base pairs of DNA from the left of this interval to make the left flank, and amplifying 500 to 1,000 base pairs from the right of this interval to make the right flank. And so when I say amplify, we will have to do PCR. Now let's take just a closer look at what the PCR for the left flank would look like. Okay, so typically we need 7 reagents for a PCR reaction. So we need the DNA template, which is going to be the genomic DNA from RDGR 170.3. So we need that genomic DNA. And you actually didn't need to isolate DNA from this strain to start this process. I gave you the genomic DNA for that. So we would need, for the left flank, for vector 375, we would need a forward primer and a reverse primer. So for this specific vector, the forward primer has a name V0375-A. The reverse primer has a name V035-B. And so all of your vectors will probably have different primer names. Okay, so components 4 through 7 are going to be the DNTPs, deoxyribose nucleotide triphosphates, the PCR buffer, the DNA polymerase, and water. Okay, so all of those reagents will go into your PCR reaction. And, okay, so what's going to happen? Well, let's say this is the genomic DNA, the template, and let's say this is the target interval that we are planning to replace with a hygromycin resistance gene. So I should change this, right? Let's go with V0. Sorry about that. I made a mistake here. I'm going to name this B, and I'm going to name this A. I had it labeled R and F for some reason. That was wrong. Okay, so these are the primers. Notice the primers bind and amplify the left flank of this region. Okay, so PCR, template denatures, primers anneal, and after many, many rounds of PCR, 25 to 30 cycles, say, we will have about a billion, I should say, molecules of the left flank. So this is what we tried to amplify. We have the primer that goes, the reverse primer, the forward primer. After about 25 to 30 cycles, we will have about 1 billion molecules of the left flank. And that's just for the left flank. We would need to do a similar thing for the right flank. Now, one thing I need to add is that we have a slight twist in the procedure used to amplify the left flank and the right flank by PCR. So for the left flank, the reverse primer has what we call a 5' extension. So let's say this is the target region this way. I'm just showing a few base pairs of that target region that we want to replace with the hygromycin resistance gene. And I'm showing to the left, I'm showing DNA to the left of that region over here, at the left. Now I'm showing the DNA denatured, and I'm showing the reverse primer for left flank amplification annealed to the DNA. And so DNA polymerase would come in here and extend this 3' end that way. I'm not showing primer binding to the opposite template strand. I'm just focused on this one reverse primer binding to the template. And I'm focused on the reverse primer because the reverse primer has this weird extension. So let's say how many base pairs of this primer anneal to the DNA, the template DNA. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So 20. Okay, so 20 base pairs anneal to the template. And you can see this is the border of the left flank and the target region. So that's great. The primer anneals perfectly to these sequences. So these blue bases here, they don't match the target region. They do not anneal to the DNA because they are not complementary to any sequences in the genomic DNA. these are going to be complementary to base pairs in the center fragment. They are going to allow us to fuse the left flank to the center fragment by PCR. And usually for our extensions are about 25 to 30 nucleotides long. So you can think of this reverse primer for the left flank as sort of a hybrid primer, where half of the primer anneals to the template and the other half towards the 5' end does not anneal to the template. So we do a similar thing for the right flank, and let me just go over that with this slide here. Okay, so for the left flank PCR, the forward primer is normal. And what I mean by normal is all of the base pairs, all of the nucleotides, not base pairs, all of the nucleotides in the forward primer will anneal to the template. There's no differences. The reverse primer has a 5' center fragment extension. So the 5' end of that reverse primer, remember, reverse is this way, the 3' end is over here, the 5' end is over here. That 5' end is going to be an extension that does not match the genomic DNA template from strain RDGR 170.3. It doesn't match. And so after PCR, what we will have, it will have the left flank with this part in blue that matches the center fragment, and this part, which is uncolored, will be the left flank sequences that are specific to genomic DNA from the SK3 strain, from genomic DNA of strain RDGR 170.3. And let's say these are 25 base pairs. So about 25 base pairs at this end of the left flank matches one end of the center fragment. So over here, same thing happens, or similar thing happens when we try to amplify the right flank by PCR. So, however, the forward primer is the one that has the 5' center fragment extension, and we can say it is about 25 nucleotides long. The reverse primer is normal. As a result, the product of the right flank will have 25 nucleotides that match the center fragment, one of the ends of the center fragment, and then the rest of that product will match the right flank of the target locus. Okay, so these center fragment matching ends are going to allow us to fuse the left flank and the right flank to either side of the center fragment. So what about the center fragment? So you didn't need to, for those of you who have already made these vectors by double joint PCR, did not need to amplify the center fragment. I provided the center fragment to you. So the center fragment, the DNA template for the center fragment is not Neurospora crassa genomic DNA. Instead, it's plasmid DNA isolated from an E. coli strain called PTH1119.2. So this is the name of the plasmid. It's also the name of the E. coli strain that harbors this plasmid. So we isolate this plasmid from the E. coli strain, and we use this plasmid as a DNA template in PCR with two primers, one called HPH-Sen-F and the other called HPH-Sen-R. And what I'm showing in this plasmid's about 4.5 kb, so 4,500 base pairs long, and it has some important sequences. One of the sequences provides resistance to the particular strain of E. coli with the plasmid, provides resistance to the antibiotic ampicillin. But these sequences over here, now this is the hygromycin resistance gene. This is the hygromycin resistance gene. And this in blue is a promoter that works in Neurospora crassa for expression of the coding sequence of the protein that provides resistance to hygromycin. So promoter is in blue, coding region is in red. And I have that over here. So after PCR with this template and the HPH-Sen-F, HPH-Sen-R primers, we will have a 1,000 1,412 base pair DNA molecule that looks something like this, with the TRPC promoter that will allow for expression of the hygromycin resistance gene coding region in the fungus once this is incorporated into the fungal genome. Okay, so that's how we get the center fragment. And like I said, most of you did not need to amplify, did not need to perform this PCR reaction on your own. I provided you a tube containing the center fragment to fuse to your each of your left and right fragments. So many of you have unique left flanks and right flanks, but all of you use the same center fragment, the same sequence, the same 1,412 base pair molecule. So review. Now, in order to construct the vector, we need to do a PCR reaction for the left flank, a PCR reaction for the right flank, and a PCR reaction for the center fragment. Now, the templates for the left flank and the right flank are RDGR 170.3 genomic DNA. The template for the center fragment was plasmid DNA. Now, the forward primer for the left flank was normal. The reverse primer had the 5' extension, which matches the center fragment. For the right flank, forward primer had the 5' extension that matches the center fragment. The reverse primer was normal, no extensions. And the center fragment primers were normal, no extensions on those. After you did the PCR, what you would do, what we normally do is we run the PCR products on a gel, and we extract the PCR products from the gel and we purify them using a procedure called gel purification. And most of you, if you've constructed a vector already, you are familiar with this process. So essentially what we will end up with is a left flank of about 500 to 1,000 base pairs with 25 base pairs that match the center fragment. Let's say the TRPC promoter end. For the right flank, 500 to 1,000 base pairs, and I'm giving this range because all of you have a slightly different length flank that you are using in your vectors. But the right flank will look like this, 500 to 1,000 base pairs with about 25 base pairs matching, say, the hygromycin resistance gene coding region over here. So once we have all of these, how they are normally stored is they're stored at -20 degrees after gel purification. They're stored in these little microcentrifuge tubes, which hold about 1.5 mil total. After purification, we usually have about 50 microliters of solution containing each of these different DNA molecules. So we have a tube for the left flank, a tube for the right flank, and a tube for the center. Now each of you for your different vectors will have probably different left flanks and right flanks. Sometimes the right flank or left flank is similar between vectors, but no two vectors have both left and right similar to each other or identical to each other, but the center fragments are always identical. So to fuse these fragments, what we use is the double joint PCR procedure. And where does this name come from? Well, the joint 1 can be considered to be just the fusion point between the left fragment and the center product. So that's joint 1. Joint 2 is just the fusion point between the center fragment and the right flank. So we use the fusion PCR step to fuse these together. And the reason it works is because there is 25 base pairs of matching sequences between these molecules over here and over here. I'm going to show you why that works in a moment. After the fusion step, now this isn't a very efficient process. So we don't even check to see if this worked by gel electrophoresis because so few molecules fuse. It would be difficult to detect this on a gel. So what we typically do is just take the fusion product from the fusion PCR reaction. Notice there are no primers. There are no extra primers added to this. The primers, as I'm going to show you, are built in to these products. I'll show you that in a moment. But because this is so efficient, what we need to do is add primers after the fusion event. After the fusion event, we take a little bit of the product from the fusion step. We add some what we call nested primers, which bind inside, and we do a regular PCR reaction with 25 to 30 cycles, and we end up with a slightly shorter product that has slightly shorter flanks than what we started with. The center fragment is unaffected. It's just we shrink the left flank and the right flank a little bit based on our choice of internal primers for the nested amplification step. And then we purify this using column purification. That just gets rid of the enzyme, the DNTPs, and that's what we use to transform Neurospora. We have a left flank that matches the left side. That matches the left side of the DNA interval we wish to replace with this hydromycin antibiotic resistance gene. And we have the right flank here, which matches the right side of that interval we wish to replace. So let me go over a little bit of how this fusion step works, because this is important, especially for those of you who are doing posters and seeing your thesis. You really need to be able to show you understand how this process works. It's such a critical step to your projects. So, okay, this is what I wanted to do here. Am I still recording? I hope I'm recording. Yes. Okay, so these are the diagrams I have been using for the left fragment, the center fragment, and the right fragment. I'm going to use some conceptual diagrams down here. These are conceptual also, right? But let me use some others. So I'm going to use this type of diagram. So this left fragment is going to be represented by these 10 base pairs here. This blue region, instead of being 25 nucleotides long, is going to be represented by just these 3 base pairs down here. So instead of 25, I'm just going to use 3. This is the center fragment. The center fragment is going to be represented by this right here. So we have the three base pairs that match these 3 base pairs. See that? Okay, these 3 match these 3. Normally, there'd be 25 here that match 25 over here. So this is the right fragment. These 10 base pairs here are representing the 500 to 1000 base pair long right fragment. It's being represented by these 10 base pairs. This 25 nucleotide base pair region right here is being represented just by these 3 base pairs here. And see how these match these 3? Normally, this would be 25. These would be 25 base pairs long. These would be about 25 base pairs long, and they match. Okay, so hopefully this little, well, I'm going to walk you through how this process works just by using these 3 molecules here. So how the fusion process works. So let's see. Let's say we mix these 3 molecules in a PCR reaction. We don't add any primers, so we leave out primers, we add extra water. So we have these molecules, we have water, we have DNTPs, we have buffer, we have the enzyme. So at the denaturation step, all of these molecules denature. So we're left with something that looks like this. Let's see, I have left top, I have left bottom, I have center top, I have center bottom, right bottom, and right top. Now there are no other DNA molecules in the PCR reaction, at least there should not be. We purified all of these molecules by gel purification, and we only added these DNA molecules to the PCR reaction. Now when we lower the temperature of the reaction to the annealing step, some of these molecules will come together because of the matching ends, right? So let's take just the left top and the center bottom. So that would be the top of this left fragment here and the bottom of the center fragment. Now you can see I can add 5 primes here, 3 prime here, 3 prime here. You can see that these are complementary to one another at the 3' ends. And normally, this 3 base pairs, that's too short for these to stick together. They would be 25 nucleotides long in practice. But for this conceptual exercise here, let's assume these 3 base pairs represent 25 base pairs, and they are complementary. So this is at the annealing temperature. Now when we raise to the extension temperature, DNA polymerase can come on here and extend this 3' end all the way out to here. And it can also do the same thing down here, extend all the way out to there. So after those have been extended, we have something like this. So the left fragment has been fused to the center fragment. The left fragment has been fused to the center fragment. Now, maybe I can get a clean piece of paper for this right here. So let's say, and some of the other molecules we're fusing too, but I just focused on the left top and the center bottom for that first step. Now, after the polymerase extends those 3' ends, we have a fragment with consisting of the left flank and the center fragment. Now, let's say all the molecules are denatured again at the denaturation step. Let me put the right top and the right bottom back in here. So these get denatured. So here we have the denatured fragments of the left top, left bottom, center bottom, and the right top and the right bottom, and the other fragments are still in here, but we really only need to focus on these to see how the fusion steps are now going to fuse the left and the center to the right. Okay, this is the denaturation stage. Now we lower to the annealing temperature, and these can come together like that. Let's see. That looks pretty good. So this one's not going to do that much, right? Because we have 3' ends that aren't attached to any template, so we can get rid of that one for now. But look at right here. Left top, center top is now annealed to right bottom. Now remember these 3 base pairs here in practice would be 25 base pairs. There'd be 25 base pairs of complementarity between these sequences. So at the extension temperature, DNA polymerase can come in and extend this to the end of the right bottom fragment and can extend this all the way to the end of the left top fragment. And what does that give us after extension? Well, it gives us the complete vector, right? The left flank has been fused to the center at the positions of overlap, and the center has been fused to the right at the positions of overlap. Okay, so that's fusion PCR. That's fusion PCR. And so now there's one more step. So this process is very inefficient. So let's say let's say we had millions of the left flank, the center fragment, and the right flank. Maybe only a few hundred actually fuse the way we want them to. So that's not enough to do a transformation. So researchers have found that the best thing to do is after you have fused the left flank to the center into the right, the left flank to the center fragment to the right flank, what you should do is take a little bit of that fusion product and do another PCR reaction, this time with primers that bind internal to the end of the left flank and internal to the end of the right flank. So assume we've added some primers that do just that to a PCR reaction with this as the template. We denature and then we anneal those primers. So in this case, I have a nested reverse primer that binds in just a little bit in here. And I have a nested forward primer that binds in a little bit from the end over here. So as a result, after many rounds of amplification, let's say 30 PCR cycles, we're going to lose these 2 base pairs off the end. And it may be, you know, if this was in practice, it may be 100 to 200 base pairs off each end. And researchers have found that nested amplification with nested primers is much more efficient than trying to use primers that bind on the very ends of the right and the left flank. For whatever reason, there could be multiple reasons why nested primers work much better than primers that bind all the way at the ends of this molecule. So at the end, we have enough DNA. We have enough DNA to use in transformation of Neurospora crassa. The center fragment here replaces the target interval, and the left fragment and the right fragment is just slightly shorter than the left fragment and the right fragment that we initially amplified to construct the vector. Wow, okay, that was a lot. I hope you understand that. This is especially important for those of you who will be defending your senior theses later this spring. Please watch the video, take the quiz, and let me know if you have any questions.